to start the recording again. All right, and let me share my screen one more time so we can just keep going with the PowerPoint and with the outline. So I was starting to say that this next section is what's covered on one of the pre-recorded lectures that I've posted. I've got 3 different ones. You have a lot this time because I wanted to make sure I covered everything that I could, especially the hard stuff. So for this whole section, that's regulation of glomerular filtration rate and blood pressure. If you're looking on the outline, there's a lecture. First of all, there's a lot filled in for you in the outline already. And then there's a lecture to explain it a little bit more that is posted in Unit 5. So make sure you check that out. Skip through all of that. So I have a separate lecture for the intrinsic controls. And then if you scroll down, like I'm doing, there's a separate lecture for the extrinsic controls. Keep going. And then once you get to the tubular reabsorption section, that's where the recorded lecture stops. So that's where I'm going to pick up here. Let me make sure I'm at that section myself. Okay, and let me get there in the PowerPoint. Okay, here we are. So we just talked about filtration, which is the first process of urine formation. And now we are going to get into the other 2 processes, reabsorption and secretion. So the next one that happens is tubular reabsorption, which is the movement of solutes and water from the filtrate back into the blood. Because remember, if you think initially what filtrate is, it's like everything from your blood except for blood cells and proteins. So there's a lot that initially gets filtered out of the blood that you don't want to lose in urine. These are substances that you want to keep in the body, you want to keep in the blood. So these are things that get reabsorbed. And reabsorption occurs along the entire section of the renal tubule, but especially in the PCT, which is that very first section here. Substances can be reabsorbed through either passive or active transport. Active transport requires energy. Passive transport does not. There is what's called a transport maximum for just about every single substance that gets reabsorbed within the kidneys. And there's one thing, or just a description here that I'll have you fill in the space that's there in your outline. This is a measure, the transport maximum is a measure of the maximum amount of a nutrient that can be reabsorbed in 1 minute.
I'm sorry, I've lost you. Where are we writing this in at?
At the transport maximum, it's right underneath of reabsorption. Do you see that?
Okay, yes. Okay. Can you repeat what you just said? I'm so sorry.
That's okay. So it's the maximum amount of any substance or nutrient that can be reabsorbed in 1 minute. Basically, your like transporters, these protein channels, they can only work so fast. So there's a certain amount of of whatever substance, every substance, that can be reabsorbed in 1 minute. But if you have more than that in the filtrate, it just can't get reabsorbed, because these things, like I said, they can only work so fast. They can only do so much at a time. So the transport maximum is kind of a measure of that. It's the maximum amount of any substance that can be reabsorbed in 1 minute. So I'm going to just go through the major substances that are found in filtrate that get reabsorbed one at a time, and just talk about how they get reabsorbed, starting with sodium. Sodium is the major ion that's found in filtrate. You have tons of sodium in your blood, tons of sodium that ends up going in the filtrate. So it's the most abundant ion, and it's the thing that gets reabsorbed first. So there's actually sodium potassium pumps all along the tubule, and they transport sodium out of the filtrate. It requires energy to do that. So it's pumping sodium out of the filtrate back into the blood. Then reabsorption of other ions and nutrients like glucose. Other solutes get transported out of the filtrate at the same time as sodium, also using active transport. And these other solutes include just other ions like potassium, chloride, vitamins, amino acids, and glucose. Those things will always get reabsorbed too. And then another thing that definitely gets reabsorbed is water. And water just gets reabsorbed through osmosis, which is a passive process where water is just diffusing from where there's more of it to where there's less. So it just goes from the filtrate back into the blood. Okay, there's two kinds of water reabsorption: obligatory water reabsorption and facultative. Obligatory water reabsorption is dependent upon the movement of sodium. And this is how about 90% of water gets reabsorbed. Okay, so basically, remember, sodium goes first. We said that sodium is the first thing that gets reabsorbed, and then water follows. Pretty much like as a rule in the body, wherever sodium goes, it pulls water with it. So water follows the movement of sodium. And that is always going to happen, always. Then there is facultative water reabsorption, and this is dependent upon hormones. There are several different hormones, which we actually just skipped over that section because I have the recorded lecture all about these hormones. There's 3 of them that are listed: aldosterone, antidiuretic hormone, and atrial natriuretic peptide. So you will, if you watch that recorded lecture, you'll learn all about those hormones and what they do. But basically, they influence the movement of water, based on whether or not they are secreted. So these hormones get secreted or not secreted based on the body's needs at that time. So, for example, if you are dehydrated, you are going to want to keep and reabsorb as much water as possible so that it remains in the body and you're not losing even more water through urine. So through facultative water reabsorption, your body would release certain hormones which cause even more water to get reabsorbed. Okay, so this is just dependent upon the body's needs at the time. If you don't need more water to be reabsorbed, then those hormones just won't be secreted and this won't happen. All right, so it just depends on what the condition of the body is at that time, whether or not these hormones get secreted and whether or not there's any additional water reabsorption. Okay, so reabsorption occurs primarily in the PCT, the proximal convoluted tubule. And in your outline, you can just fill in the percentages of like the various substances that get reabsorbed here. So here in the PCT, 100% of glucose, amino acids, and lactic acid are reabsorbed. Those are all things that are very, very useful for the body, either for energy or for making new structures, like the amino acids are used to make new proteins. Because they're so valuable, you want to reabsorb all of them that you possibly can. You don't want to just have them come out in urine. You want to keep them. So all of those substances get reabsorbed. About 80 to 90% of bicarbonate ions are reabsorbed. That's HCO3. So that's up here, HCO3. You can see, so what's circled here is mainly what's getting reabsorbed here. You can see glucose and amino acids, bicarbonate. And then the 65% is for water, sodium, and potassium. About 65% of water, sodium, and potassium are reabsorbed here. Okay, and then finally, about 50% of chloride ions and urea are reabsorbed here. Remember that urea is a byproduct of protein metabolism. So urea gets reabsorbed. So that all of that happens in the PCT. There's a lot of reabsorption that happens there. Then there's a little bit more in the next part of the tubule, which is the loop of Henle. The two sides of the loop reabsorb different things though, and that's important to keep in mind. The descending side, where filtrate is flowing down, that side absorbs water, maybe about 15% of the water that gets reabsorbed happens here. And then on the other side, this is where ions get reabsorbed. So coming up the ascending limb, about 30% of sodium, potassium, and chloride ions are absorbed. Okay. And then finally, reabsorption in the last part. So remember, the last part of the tubule is the DCT and the collecting duct. So reabsorption here depends on the body's needs at the time. If you, for instance, are dehydrated and you need to reabsorb even more water, it's going to happen here. If you're not, and you've already reabsorbed in the other areas of the tubule all the water that you need, then nothing is going to happen here. Nothing at all is going to happen in the DCT and the collecting duct. So it just depends on the body's needs at the time. And remember, it's dependent upon hormones, the 3 hormones that I mentioned a second ago, and they're listed again here. It's aldosterone, antidiuretic hormone, and atrial natriuretic peptide. The first 2 are secreted due to low blood pressure. Okay, aldosterone and ADH, antidiuretic hormone, are secreted due to low blood pressure. So if you have low blood pressure, what do you want to do? You want to raise it, bring it back up to where it should be. And one way of doing that is to increase your blood volume. By increasing blood volume, you're going to increase your blood pressure. So this is what's happening with the release of these 2 hormones. When they get released, you can fill in well, let's do aldosterone first. When aldosterone is released, it increases the reabsorption of sodium and then water as well. Because remember, water follows sodium. So aldosterone increases the reabsorption of sodium and water. So if you're reabsorbing more water, it's going into the blood, it's going to increase blood volume, which is going to increase blood pressure. And that solves the problem. Remember, it was triggered by low blood pressure. So now by reabsorbing even more water, you're raising blood pressure, which is what you want to have happen. Antidiuretic hormone does something very similar, except it just doesn't cause the reabsorption of sodium. It only causes increased reabsorption of water. So when ADH is secreted, it increases the reabsorption of water, which does what we just said. It increases blood volume, which increases blood pressure. And then the third hormone does the opposite, because your blood pressure isn't always low. What if you have really high blood pressure? You got to have a way of fixing that too. So there's atrial natriuretic peptide. It's secreted due to high blood pressure. So it inhibits the reabsorption of sodium and water, which decreases blood volume. And as a result, it decreases blood pressure. Okay, so hopefully that makes sense. We've got two hormones that will increase blood pressure due to the reabsorption of water, and you have one that does the opposite. It decreases blood pressure because it inhibits the reabsorption of water. Okay, so we've now covered reabsorption, and the last step in urine formation is secretion, tubular secretion. You have actually all this information written out in the website where I post the videos and the like written stuff, but I'll give you a couple quick definitions here. And then we'll get to, I do want to spend a little bit of time on the other outline. We're almost done with this one. So secretion involves the movement of substances from the blood into the filtrate, maybe things that didn't get filtered initially. So there's a few important functions of secretion: to remove substances that are attached to plasma proteins. There are certain things that attach themselves to proteins. So because these proteins are too large to be filtered, the substances that attach to them will also not be filtered. Okay, a main example of this would be like drugs or medications, antibiotics. They attach to the large proteins in your blood. And because those proteins get filtered, these don't either. So if there's too much of a drug, if it starts to build up in the blood, it gets secreted into the filtrate. So that allows you to get rid of it. Another thing is to eliminate waste products that have been reabsorbed by passive processes. So as we've discussed, there's some substances that are just, no matter what, some things are automatically reabsorbed. You can't help it. They get automatically reabsorbed. And what if there's just too much of them in the blood? You have to have a way of getting them out of the body. So you would then secrete them in order to get rid of them. These are things like urea. Remember, we talked about how urea does get reabsorbed, but it's a byproduct of metabolism. It doesn't really serve a function. You want to get rid of it. So it does end up having to be secreted. So it goes back in the filtrate. Okay. Also, to get rid of excess potassium. Almost all the potassium ions that get filtered are later reabsorbed. And you might not think that's a big deal, but potassium ions, if you have too much, it can actually be harmful. It can, it has negative effects on the heart. It can cause an irregular heartbeat. So you don't want too much potassium building up in the blood. So if that's starting to happen, because a lot of the potassium gets reabsorbed, it can be secreted, which means you're just putting it back in the filtrate so that it comes out in urine. And then finally, to control blood's pH. So hydrogen ions and bicarbonate ions are the two main ions that can be secreted in order to maintain the proper pH of blood. Remember, you don't want it getting too acidic. You also don't want it getting too basic. Okay, and that takes us to the end of this outline. So we got through all of urine formation. If you guys have the other one or print it out, or you can pull it up on your computer. I'm pulling it up right now. We'll get into this one. I do have a recorded lecture on, let's see, this next section. I'll introduce, let me see. I'll give you guys like a quick definition for a couple of things, but I don't want to spend too much time on it because I have a lecture for that first part. So then I'll skip down to the stuff that I don't have a lecture for. So, okay, I'm looking at the outline. The whole first section is regulation of urine concentration and volume. So there's a whole lecture on this. I'll give you guys a quick definition for osmolality because I'll use that term a lot. The term osmolality measures the amount of solutes in body fluids. Okay, but I do talk about that in the lecture, and I'm not going to do much more with this section because you have that recording. I don't want to waste our time on that. So let's see, let's skip to the tests of kidney function. Just go all the way down, scroll past the diagram, production of dilute urine, production of concentrated urine, and then there is the tests of kidney function. Okay, so one of the main things that's used to test kidney function is called a renal clearance test. And for the definition of this, renal clearance is basically the volume of blood plasma from which a substance is completely removed per minute. Okay, and in other words, this test can tell you how well a substance gets removed from the blood, gets filtered out of the blood and excreted in urine. Okay, so if the renal clearance of a substance is high, that tells you that the substance is efficiently excreted from the body. So it is removed from the blood very well, and it's excreted in urine. If renal clearance is low for a particular substance, then that means the opposite. It means it is not efficiently excreted. So that means it tends to remain in the blood. That substance is not removed from the blood very well, and it's not excreted from the body. Okay, the substance that's typically used to measure this is called inulin. Don't confuse that with insulin, not the same thing. Inulin is a polysaccharide. It's just the standard thing that's used for the test. Inulin has a clearance value that is equal to the filtration rate. It has a clearance value. I'll explain what that means, but it has a clearance value that's equal to the filtration rate. Okay, so basically what that means is that we know this substance is 100% filtered. So all of it that's in your blood will get removed from the blood. It gets filtered out. And then we also know that it doesn't get reabsorbed and it doesn't get secreted. So 100% of the inulin that is put into your blood should come out in urine. And if it doesn't, that can indicate a problem with the filtration membrane. So for any substance, the renal clearance value tells you how that substance gets handled by the kidneys. Is it filtered well? Is it not filtered well? Is it getting reabsorbed? Is it getting secreted? So it can tell you that type of information. If the clearance value is equal to that of inulin, then you know, just like inulin, you know the substance is not reabsorbed or secreted. So all of it gets filtered and then it doesn't get reabsorbed. It doesn't ever get put back into the blood, just stays there and it comes out in urine. If it has a clearance value that's less than inulin, so if there's less of the substance coming out than what you put in, then that tells you, or than what you started with in the blood, that tells you that the substance is being reabsorbed. That would explain why there's less of it in the urine than there is in the blood, because it's getting reabsorbed. It's being put back into the blood. If the renal clearance is 0, so that means there's none of this substance is coming out in the urine. And that means one of two things. That means either the substance is completely reabsorbed. So 100% of the substance that got initially filtered out of the blood was reabsorbed by blood. So there's none left in the filtrate, and therefore none coming out in urine. Or it could mean that the substance was not even filtered in the first place. Maybe it couldn't go across the filtration membrane. So that would explain why none of it is coming out in the urine. It would have a clearance value of 0 if that was the case too. Okay, and then finally, if a substance has a clearance value that's greater than inulin, that means that the substance must be getting secreted. So it's being filtered, and then even more of it is being put into the filtrate through the process of secretion. Okay, so really the main way that this is just like the application for this information, like in real life, is that it's mainly used by pharmacists in prescribing and doctors in prescribing medication. So looking at like a drug or a medication, if renal clearance is high, remember that means it is efficiently removed from the blood. But for a drug to work, it has to be in the bloodstream for a certain period of time. Otherwise, if it's removed right away, it's not going to do anything. So if the renal clearance of a particular drug is high, that means that the dosage must be high, and the drug should be taken maybe several times a day, not just once a day, but 2 or more times a day. And that's just to maintain an adequate level of it in the blood. Okay, and then on the other hand, if the drug has a low renal clearance, which means that it is not efficiently removed from the blood, so it has a tendency to remain in the blood, then the dosage would be low and it would only be taken once a day. Okay. For urinalysis, we do talk about this in our the wet lab that we did. I did with half of you guys on Tuesday, and that I'm going to do with the other half. I'll go through the things that we didn't discuss during the lab, the kidney dissection lab. So actually, yeah, we didn't talk about normal volume produced per day, about 1 to 2 liters per day. The odor, so I mean, generally it just should be like a non-offensive odor, not super strong. We did kind of talk about the color and the transparency. Fresh urine should be clear and yellow, but it's going to vary on the shade of yellow. This shows you the various shades that it could be depending on how hydrated or dehydrated you are. It always is going to be some shade of yellow though, and that is due to urochrome. That's a pigment produced during the breakdown of hemoglobin. Okay, and whenever you have more concentrated urine, it's going to be like a darker color. So the more concentrated it is, the darker the color. The pH typically ranges between 5 and 7.5. The average would be about 6. If you have, so, and we did talk about this, it depends on, the pH really tends to be acidic because from metabolism, you are producing acids as a result of normal metabolism. Also, though, our diets tend to be acidic. Think about like proteins contain amino acids. So if you do have increased protein in your diet, it does tend to be on the more acidic side. If you have less protein, which often comes with a vegetarian diet, it would be more alkaline. Another thing that's measured, we talk about this in lab, is the specific gravity, which tells you the weight of a volume of urine. Compared to the weight of an equal volume of distilled water. So if you're comparing urine to distilled water, and distilled water gets a value of 1, urine is going to be a little bit higher because urine is water, but with some added solutes in it, like ions, various molecules. So the normal range for the specific gravity of urine is 1.001 to 1.035. And if you have a higher specific gravity for urine, it just tells you that there's more solutes present, more solid particles present. Okay, looking at the chemical composition of water, you guys can see the breakdown here on the PowerPoint slide. It's about 95% water. The other 5% are solutes, which are mostly ions, but also could be various organic molecules. Like, I gave you the 3 main examples here. Urea, remember that's a byproduct of amino acid breakdown. The next one is uric acid, which is a byproduct of nucleic acid breakdown. So remember, nucleic acids are DNA and RNA. So if those DNA and RNA are being broken down, it's going to produce uric acid. And creatinine. This is a byproduct of the breakdown of creatine phosphate. Remember, creatine phosphate is a molecule that is used even before glucose. It's used for energy, but we don't have a lot of it, so it doesn't provide a whole lot of energy. So those are the main organic molecules, and then the ions, major ions are listed here that are found in urine too. All right, let's go through this table really quickly. We talk about a couple of these in lab. So if you see certain substances in urine that are abnormal, it can indicate certain conditions and diseases. So all the things that are listed should not be present in urine. If they are, you can see the name of the condition and the possible causes. So I do know that on your test for this, I do have a matching section with this. So it'll say like the substance and maybe the name of the condition if you have that substance in urine, or it might do it like the substance or the name of the condition and then the cause, and you're just matching them up. So for glucose, possible causes of having glucose in the urine, the big one is diabetes. Okay, I don't want to get into too much detail right now because we talk about this in lab. So you guys that have already had lab, you know why. I've explained why. If you haven't had lab yet, I will do that when I see you guys on Tuesday. Proteins, if you have proteins in urine, that could be caused by hypertension. That's high blood pressure. It could also potentially be caused by renal disease if you have kidney disease. Ketone bodies or ketones. Causes could be starvation, certain diets, like a really low carb diet. And you can also see this sometimes with diabetes if you're not treating it or if it's undiagnosed. Bile pigments. If you're seeing bile pigment like bilirubin, then that can indicate liver disease or an obstruction in the bile ducts. Okay, erythrocytes are red blood cells. So basically that's blood in the urine. Causes would be most likely physical trauma. I gave the example in lab that this is often seen with like a kidney stone that causes physical trauma to the inside of the kidney or the ureter. And then also leukocytes, those are white blood cells. That is most often caused by a urinary tract infection. Okay. Chronic renal disease is the next thing here. Basically with this, the glomerular filtration rate gets really low. Glomerular filtration rate will go to less than 60 milliliters per minute. So it'll be less than 60 milliliters per minute. And just so you have a comparison, normally filtration rate should be over 70 milliliters per minute. Okay, so think about how that would negatively impact the body. If filtration rate is not as high as it should be, filtration isn't happening as quickly as it should, then that means wastes are going to build up in the blood. I mean, the biggest issue that's going to cause is that it's going to lower the pH of blood. And that right there could cause death if it lasts for long enough and if it gets low enough. Often chronic renal disease is caused by, it can be a bunch of things, but common causes are diabetes and hypertension. And then someone would be considered to be in renal failure if the filtration rate drops to less than 15 milliliters per minute. That's very low. And that gets really dangerous. So at that point, the person would likely have to be on dialysis. Okay. So we really focus just on the kidneys. I mean, they do the majority of the work of the urinary system, but we have the other structures as well. You guys know the ureters are the tubes that carry urine out of the kidney. For the histology, there's actually 3 layers of tissue in the ureters. The mucosa is the inner lining. It's transitional epithelium. Transitional, you guys may or may not remember this from A&P1, but if you have transitional epithelium, it actually changes shape due to stretching. So the ureters can actually stretch to accommodate urine as it passes through, and the cells change shape. The muscularis is just a layer of smooth muscle. And the adventitia, the outer layer, is tough, fibrous, connective tissue. Okay. Sometimes kidney stones can develop in the ureters, or often they're in the kidney itself. And these are solid, basically solid crystallized clusters of dissolved minerals. So it's like a solidified, crystallized cluster of minerals that were in urine, but they've crystallized and hardened. And they often develop in like somewhere in the calyces, the renal pelvis, or in the ureter. And they can cause an obstruction. They can be really painful. So symptoms are definitely pain in the abdomen, pain in the groin, and having blood in the urine. Okay, for urinary bladder, it is a hollow muscular organ that it serves as a storage area for urine. And it has the exact same 3 layers of tissue as the ureter. So you can fill in the same thing: transitional epithelium, smooth muscle, fibrous connective tissue for inner, middle, outer. One special thing I'll tell you, I'm going to type it in. There is a name for the muscle layer in the bladder. It is called the detrusor muscle. That's the name for the smooth muscle layer there. Okay. The trigone, I point this out in lab when I do the model, the urinary system model. It's just a triangular area at the base of the bladder. Okay, and I'm going to end once I get through the urethra. Do know that although I'm not quite getting to that last section, it's not very big. It's just 2.5 pages of body fluid composition. You have a whole table in the web page that I've created that basically allows you to fill almost all of that in. So you'll have pretty much all of that information if you go to the web page that's in the second Unit 5 section in D2L. All right, so the urethra, real quick, I've got 2 minutes. The urethra is just the tube that extends from the bladder to the outside. There are 2 sphincters. Remember, a sphincter is a circular muscle at an opening. So 2 sphincters associated with the urethra. The first one is involuntarily controlled. It is smooth muscle, so it automatically is going to open and close. But the external sphincter is voluntarily controlled. So you determine when that opens and closes. And the term micturition or micturition reflex, that refers to urination, the discharge of urine. Your bladder capacity is about 800 to 1,000 milliliters, but often it's not filled to capacity. Once you get to not even half capacity, the pressure in the bladder increases enough that it's detected by stretch receptors, and action potential is sent to the brain, and that gives you an awareness of the need to urinate. So that makes you aware. And involuntary action potential will relax the internal sphincter, because remember, that's involuntarily controlled. But then you decide when the other sphincter gets opened. So a voluntary action potential will relax the outer external sphincter. And that's when the bladder will empty. Okay, I finished just in time. It's 11:20. So, all right, we're all done with the outline. Please make sure, because I have several pre-recorded videos for you guys. please make sure you watch those, because the three, especially in the first part of Unit 5, are going to explain some of the physiology of regulating the filtration rate and regulating blood pressure. So make sure you watch those, and then check out the webpage where those videos are, to fill in any parts that we haven't gotten to. We got to almost everything today, which is good. You should be able to fill in everything else, but obviously let me know if you guys have any questions about anything. I'm going to stop sharing, stop the recording, and